Um, my name is Robin Pryor. I am one of Javed Shah's graduate students, and I'm introducing Dr. Dan Barber, who is a chief of the T lymphocyte biology section at the National Institute of Allergy and Infectious Disease. Dr. Barber obtained his bachelor's from Rider University and his PhD from Emory University in the Department of Microbiology and Immunology. Afterwards, he joined the Laboratory of Parasite Diseases at the NIH as a postdoctoral fellow in the immunobiology section. He was awarded a position as an Earl Stadtman tenure-track investigator in the Laboratory of Parasitic Diseases. His lab performs research on the PD-1 pathway in CD4 T cells during Mycobacterium tuberculosis infection. So I think the way this is set up, if anyone has a question, you can post it in the chat and I'll read it. I think that's the setup that we're going to go with. So Dan, do you have a preference in terms of questions during the talk or after? Are you fine with both?
Absolutely fine with both. I like to be interrupted because while questions are fresh in people's minds.
All right. So you can go ahead and start.
Okay, great. Waiting for this to power up. Can you guys see my screen? Okay, great. Thanks, Robin, for the invitation and for that introduction. I'm really happy to talk with you guys today. So, as Robin mentioned, our primary interest is in T cell responses to TB. And today I wanted to show some mostly unpublished data and a little bit of recently published data. We're trying to take very different approaches to asking the questions whether or not we can manipulate T cell function in a way that may be therapeutically beneficial in the context of MTB infection. And first, I'll talk about our work on the role of the co-inhibitory receptor PD-1 and the role that plays in MTB infection. And then I'll show some data from a newer direction in the lab and trying to understand the role of mucosal associated invariant T cells or MAIT cells in the context of TB infection. And I'll show some recently published data in mice and some unpublished data from macaques. I think it goes without saying that there's really a great need for the development of novel prophylactics and therapeutics to address the TB problem. The vaccine we have for TB is 100 years old and doesn't work great in preventing tuberculosis in adolescents and adults. And so the development of a highly effective vaccine for TB would have an enormous impact on the global pandemic. We also really need to develop novel antibiotics, in part due to the problem of antimicrobial resistance, but of course, TB chemotherapy is long, and novel antibiotics, which could shorten treatments, would really have a huge effect as well. But what you hear discussed a lot less frequently is the idea of development of host-directed therapies, the idea that you might be able to manipulate inflammation in the host in a way that's beneficial to the outcome of infection. Now, I think the role for host-directed therapies will primarily be as adjunctive, so something that you could co-deliver along with antibiotics to sort of reduce treatment or reduce the relapse rate. So some sort of way of enhancing immunity along with actually attacking the bacteria itself. And our main interest is, as I said, is trying to understand if we could manipulate T cell function in a way that enhances control of the infection. So that really the center of our tuberculosis immunological universe is the CD4 T cell and macrophage interaction. We know that CD4 T cells are absolutely required for control of TB, but so often TB infection results in a persistent infection. While you have what appears to be an ongoing and effective CD4 T cell response, it doesn't lead to elimination of the bacteria. And so our question is can we manipulate CD4 T cell or CD8 T cell responses in a way that are important or that enhance killing of the bacteria? Now, the most obvious candidate when you start talking about manipulation of T cell function is the co-inhibitory receptor PD-1, which I think most people are familiar with now. But very briefly, PD-1 is a cell surface molecule that's induced on T cells upon TCR signal transduction And when it interacts with one of its ligands, PDL1 or PDL2, recruits inhibitory phosphatases to the plasma membrane, and it results in an attenuation of signal transduction to the T cell receptor, and just as importantly through C28. And when you block the interaction between PD1 and one of its inhibitory ligands, that results in enhancement of function of T cells. And this has been really translated to enormous success in immuno-oncology. And there's, as you know, I'm sure, there are a number of FDA-approved drugs now for the treatment of various different malignancies. There's a number of different settings where disrupting the PD1, PDL1 interaction boosts T cell function in such a way you can get pretty dramatic tumor reduction. And so this is a major new avenue for, it's not so new anymore, for treating cancer. And in fact, this led to the 2018 Nobel Prize to Jim Allison and Tasuku Hanjo for the development of checkpoint blockade immunotherapies for cancer. And so this has, of course, led a lot of us in other fields to think, well, can we also apply what we understand about PD1 and T cell exhaustion and boosting T cell function through different PD1 blockades to the treatment of other settings, and in particular tuberculosis. And so the idea has been out there and persisted for quite a while that maybe we could get just as good of an outcome with TB as one might see with cancer by blocking the PD1 pathway. However, the first data that came out about the role of PD1 and TB came from mouse models, first published by Bill Jacobs and then not longer by Alan Scher and I, where we found something quite unexpected, and that is that PD1 deficient mice are horribly susceptible to Mtb infection. And this is really I think the first and maybe still the only example of removing a negative immune gene, a negative regulatory pathway, leading to severe host susceptibility. Normally you think of a knockout mouse being deficient in T cells or interferon gamma or IL-1 or TNF for dying of TB. Here we removed an inhibitory receptor which boosts T cell function that led to the early death of the host. And we were able to go on to show that T cells themselves were actually largely responsible for this rapid death. CD4 T cells primarily, and to a lesser extent, CD8 T cells. We still don't know, excuse me, we still don't know much about the mechanisms behind T cell exacerbation of tuberculosis in the absence of PD1, but we have gone on to show at least a little bit of data that when you block PD1, interferon gamma production by T cells goes up quite a lot. And rather than enhancing control of the infection, that leads to a lot of cell death in the lungs, necrotic lesions, and eventually a dead mouse. And so blocking the PD1 pathway in TB is doing what you'd expect. It's enhancing T cell function and making the Th1 cells make more Th1 cytokines like interferon gamma, but in the context of TB, that actually makes things worse instead of making things better. So the thinking from the mouse model data was that you need negative regulation through PD1 to alleviate or to prevent detrimental inflammation. And that a little bit of negative regulation of T cells is important for host resistance. Now, as I mentioned, there's a lot of people being inoculated with anti-PD1 blocking agents for cancer immunotherapy. So you might think that there'd be cases of tuberculosis associated with cancer immunotherapy. And in fact, over the past few years, there have been an increasing list of case reports of TB that occur in patients after starting PD1 blockade. And in fact, even the product label of atezolizumab lists the tuberculosis cases that were seen on trial. And every single class of PD1 blocking drug so far has had cases of tuberculosis reported to the FDA. So there's a little bit of a hint here, actually, even though there's very little immunology, or very little immunology, I should say, or almost none done on any of these patients, any of these case reports. So there's a hint here from the literature that maybe the mouse data is correct. The problem, however, is that the mouse model data is from mice, and mice are really, quite frankly, hit or miss as to whether or not they are predictive of what would happen in the context of TB in humans. And the problem with this data here, the clinical literature, these are individual case reports, and TB is kind of everywhere anyway. So it's hard to know if this is really a signal. and that there is a causal relationship between the cancer, between tuberculosis in these patients and PD-1 blockade. So we thought that we really wanted to address this question. Maybe the rhesus macaque would, or the non-human primate model of TB would be a great way of trying to, and at least in our minds, settle this question. And so we designed the study where 6 macaques were given isotype control and 6 macaques were given anti-PD-1 starting at 2 weeks post-infection and continuing out to week 15 and 16 where necropsies happened. And so we picked 2 weeks because that's right. We wanted to let the bug take and grow initially for some time before we started the intervention. But 2 weeks is right around where T cells are first probably starting to get primed in the macaque. And so that's when we started blockade. We also picked a strain and dose of TB that we had past experience with that we knew would result in a pretty low level of disease, because we were hypothesizing that we would make things worse with PD-1. So we didn't want to have a setting where we had rampaging tuberculosis in the macaques and have it difficult to observe immunopathology on top of terrible virulence of the bug. So we picked the setting where the control monkeys are going to be essentially latently infected, even though that's a tricky thing to say in rhesus. So just a few gross clinical readouts. All of the isotype control treated macaques continue to gain weight through the study. We had one rhesus macaque in the anti-PD-1 treated animals have to be euthanized early. It developed severe symptoms and the vets made us take it down. And then there was one macaque that developed wasting disease. But 4 of the other macaques continued to gain weight. When we looked at we also followed disease in these macaques using FDG PET-CT imaging. And here I'm showing the data normalized to week 4, which is where we can first start to detect the PET-CT signal in these animals. You can see the PET signal on the isotype control treated macaques is relatively stable over the 16 weeks of infection. But we did have one macaque, or several macaques develop progressive PET-CT signal over the course. And in fact, one of the macaques hadn't lost any weight and made it to the endpoint, but actually had the highest level of disease as measured by PET-CT. And so overall, none of the control animals had any overt signs of disease. And I think we probably could have classified them as latently infected healthy animals at the end. But 3 out of the 6 animals that received the anti-PD-1 were in some sort of trouble. One was euthanized, one was cachectic, and the other had pretty good disease and was probably going to eventually start to go bad after, and not long after the study. Really, the most important data, though, for this whole study is this slide here, and that's the bacterial loads. So each at necropsy, we hunt through, we take the lungs out of the monkey and we bread loaf them, and we hunt through every square centimeter of the lung and carve out every palpable granuloma that we can find. And then each one of those is plated for bacterial loads and immunological analysis. And so each dot here on this graph on the left where it says pooled data is an individually resected granuloma from the macaques at necropsy. And so we clearly did not make TB better by blocking PD-1. There was a pretty significant increase in bacterial loads after PD-1 blockade in individually resected granuloma. So we're actually talking about bacterial replication at the actual lesion. If we break this up into the individual macaques, about 4 of the anti-PD-1 treated monkeys had bacterial loads that were higher than the highest of the control macaques, but 2 of them were just fine. And if you look, there's the spread goes all the way from just fine to really high. So there's an enormous amount of heterogeneity here. But it seems to be that in about 4 of the 6 animals, we had some sort of exacerbation of bacterial growth after PD-1 blockade, ranging from mild to pretty bad. And 2 of the macaques were just fine. So this seems to be pretty consistent with the mouse model data, is that PD-1 is actually host protective in the context of TB, and blocking it makes the infection get worse. Now, we're never really going to know why in the macaques PD-1 blockade made the bacterial loads go high, but we did a series of experiments to try and look at what happened to the immune response in these animals to see what correlated with this, what was PD-1 doing and what changed when we blocked it. And so first we looked at the kinetics of the antigen-specific CD4 and CD8 T cell response. CD4 is on the left and the CD8's on the right. And here we restimulated the cells with Cecilia and Alex's peptide megapools. They work fantastic in macaques. We can very readily detect huge precursor frequencies of Mtb-specific T cells. Now, we expected that blocking this inhibitory receptor, we'd see this big increase in the antigen-specific T cell response, or at least a big acceleration in the T cell response. But that is not at all what happened. In fact, quite the opposite. And it was particularly obvious at the CD4 T cell response. And looking at the kinetics this is in the bowel, in the graphs on the left, you can see in the green shaded region, we actually slowed the T cell response against TB in the bowel. By the end of the experiment, the percentages of T cell specific for TB were kind of similar, but we didn't increase the speed of the response. If anything, we made it slower. So that was and for the CD8s, it was a bit noisier, but there was a trend that maybe the same thing was happening. But we certainly didn't get what we expected. It was a bigger T cell response. We also looked at the T cell response in the granulomas. So again, each dot is an individually resected granuloma that we restimulated with the megapools and then did intracellular cytokine staining to look for antigen-specific T cells. And what we found was a bit surprising to us. There was really no difference in the magnitude of the CD4 T cell response before and after PD-1 blockade. In contrast, however, for the CD8s, there was a pretty significant increase. So rather than boosting the CD4 response, we wound up, like we expected, we wound up boosting the CD8 response against TB, more obviously anyway, than for the CD4s. Now, I'm not going to go through all the functional data. We did a large number of cytokines. We looked at a large number of cytokines by intracytokine staining, and we did not increase the function of the CD4s at all. In fact, one of the things that we noticed when we looked at other markers was that the biggest change we saw of everything we analyzed was a large increase in the expression of another inhibitory receptor, CTLA4. So blocking PD-1 didn't boost CD4s, and it just made them operate at higher levels of another inhibitory receptor. For the CD8s, that was quite different. There was a pretty significant increase in the levels of granzyme B expression. We did not see this compensatory increase in other inhibitory receptors on the CD8s. So we had a preferential impact on boosting the function and number of CD8 T cells as compared to CD4s when we blocked PD-1. We also went on to characterize the quality of inflammation in the granulomas with Luminex. So again, with each granuloma, we mash it and then we spin the cells down and we take that fluid off the top and clarify it and then use that for Luminex. So we call that granuloma homogenate. And so each one of these dots again is an individual granuloma we ran Luminex on. And the three cytokines that were the most preferentially upregulated after PD-1 blockade were interferon gamma, IL-18, and TNF. So Th1 associated cytokines, which really fit with the mouse data, where we've mechanistically implicated interferon gamma in the exacerbation of tuberculosis disease counterintuitively after PD-1 blockade. But to understand the data a little bit better, we sent it to Bruno Andrade in Salvador at Fiocruz, and he's a statistical whiz. And he performed a canonical correlation coefficient analysis. And I'm going to ask for forgiveness for the statisticians out there while I butcher this, but essentially this looks for it tries to understand how all the data is interconnected. It looks at how all the different analytes relate to one another as a measure of the quality of inflammation. And what Bruno found was that he could completely separate the pattern of cytokines in the I-step control and anti-PD-1 treated granulomas using this analysis. And when he quantified which of the different analytes contributed to the separation of these two groups, IL-18 really stood out, which was interesting to us. IL-18 was not only high, but was really implicated as being being highly interconnected with a lot of the other cytokines that were observed in those animals. Bruno also looked to see how the bacterial loads correlated with each of these different analytes that we measured. And so here I'm showing on the left each individual analyte versus bacterial loads for ISEP control, and on the left for ISEP control, and on the right for anti-PD-1 treated animals. And what you can see first and foremost, there's a lot more red bars. So a lot more statistically significant positive correlations between cytokines in bacterial loads. And so, in other words, the bacterial loads went up in the anti-PD-1 treated granulomas, and that really correlated with all these different inflammatory markers. And interestingly, the top candidate was IL-1 beta. So IL-1 beta in the isotope control treated granulomas had no correlation with bacterial loads, but in the anti-PD-1 treated granulomas, it was actually the highest Spearman correlation coefficient. And so this really caught our attention because these are both inflammasome generated cytokines. And so this led us next to look at inflammasome activation in these granulomas. And so this was a collaboration with Paul Baker and Barber's lab. So it turned out the best way to look at caspase activation was by Western blot. And so this is just an example blot of many more that we ran from the granuloma homogenate. So Paul found detectable procaspase-1 in every single granuloma homogenate we looked at. But what was interesting was the activated form of caspase, the P10 band, was really only detectable in a subset of granulomas. It was either positive or negative. So we did 23 granulomas from each group, and really there wasn't any difference in the percentage of granulomas that had a detectable P10 band. So 11 of 23 for isotope control and 14 of 23. So we didn't increase inflammasome activation with PD-1 blockade. I wouldn't have expected that I did, because we're talking about a T-cell inhibitory receptor. There isn't an obvious link to it activating the inflammasome. But we did go on to look to see how this correlated with bacterial loads. And so we separated the granulomas to those that had the detectable P10 band and those that didn't. So in other words, what was the CFU like in grans that had an activated inflammasome versus those that didn't? And for the control treated monkeys, there was really no difference. But strikingly, in the anti-PD-1 treated macaques, there was a very clear separation in that we only saw increases in bacterial loads in granulomas that had a detectable P10 band. In other words, we weren't increasing activation of the inflammasome, but if the inflammasome happened to be activated in that granuloma, PD-1 blockade was more likely to make things worse. At least that was the hypothesis. So we wanted to try and ask if this was really mechanistically linked. So we went back to mice just for this one experiment, and we crossed the PD-1 knockout mice to Caspase 111 double knockouts to make triple knockouts. And what you can see is in the pink line, PD-1 deficient animals succumb very rapidly. But in the green line, if we remove caspases from the PD-1 deficient animal, we can actually significantly extend their survival. And that is not something you'd normally see in the presence of PD-1 with the blue and black lines. And so we think that the Luminex analysis led to an unexpected new mechanism, and that somehow PD-1 blockade is really the exacerbation of TB upon PD-1 blockade is potentiated if there's ongoing caspase active or inflammasome activation inside the granulomas. And we don't know if this is a cell death thing or if this is IL-1 or IL-18, but there's something to do with caspase and PD-1 blockade. Switching gears here, other mechanisms that might be attributed to this. There's data that first from Brian Fife and Jeff Bluestone, and then from Ron Germain's lab, that PD-1 can actually modulate T-cell trafficking in tissues. And the idea is that when you block the inhibitory receptor, T-cells get stimulated harder and then they stop more. So we wanted to ask if we may be impacting on lymphocyte trafficking within granulomas. And so we developed this approach here on the left where we would do thick sections of a freshly resected granuloma that we then stain with antibodies and make movies of. And when we do that, it works quite well. You can see this is a granuloma looking at C11b, CD4s and B cells. And you see really nice movement with this approach. And overall, the myeloid cells are static and the lymphocytes move. But if you stare at it, you see all different kinds of patterns. You see cells moving rapidly, cells stopping and starting. You see cells that are very static. This was extremely heterogeneous. And so we needed to come up with some way of quantifying this T-cell movement behavior in a way that we could ask if there were differences between groups. And what we realized was that by plotting for each individual track or cell that was followed in the Amara software, if we plotted the mean speed versus the variation in speed, we came up with 3 different subsets of or three different types of T cell motilities. We have cells that were low in speed. I'm sorry, I don't think you can see my mouse because I can't see it. So I'm just going to do my best to describe. You have cells that are low in mean speed and low in their variation. In other words, they were moving slow and or not at all and never really changed much. And so we'd see those cells are stopped. Then we had a subpopulation of cells that had a pretty high average speed and low variability. And those were the cells that contained the highest maximum speed overlaid in the heat map. And so we'd say those cells are steadily moving. And then we had a population of cells that had a high variation in their track speed and intermediate mean speed. So we'd say those cells are stuttering. And we could actually go back into the movie and verify that that really is the behavior that the cells were observing. We observed in the movie. And so when we summarized a large number of movies, first you can see there's enormous heterogeneity. And just to make the point, these two graphs here on the left, they're from the same monkey that were imaged on the same day. So we saw a huge amount of variability, and it wasn't just one monkey all looked one way. Even within a monkey, there was a lot of variability. But overall, there was a trend, a statistically significant trend, that in the animals that received anti-PD-1, there are fewer granulomas that contain highly motile T cells.
Yeah, and we have a quick question from
please.
Nathan.
Yeah.
Why do only some granulomas have caspase activation? Do you know if these are the ones with bacteria and the others are sterile?
So we didn't have any sterile granulomas in this. So they all had bacteria. But as to why only some had it, I don't know. Kat is also fascinated by that question. And I think it'd be really fun to pursue that and try to look at what else correlates with that, if it's something to do with the cellular composition of the granuloma. But even in one monkey, you'll see some grands that have it and some that don't. Maybe it reflects the kinetics. There's a wave of caspase activation that then goes away, or maybe the bugs have to replicate to a point where it gets activated. I don't know, but that's a wonderful question. I did not expect that. And we did this a bunch of times to be certain that was the case. Thank you for the question. Please.
Is there a correlation with caseation? I think that's how you pronounce it.
Sure. We have not come up with an easy way to try and correlate necrosis in the granulomas with a quantitative measure. But in general, bacterial granulomas which have high bacterial loads, they're caseous. And I can just tell you anecdotally that was the case here. The granulomas from the anti-PD-1 treated macaques are physically larger. We actually measured them on a ruler, and they were bigger and mushier. And as you can see, harder to cut because we got fewer movies because a lot of them were necrotic and they just mushed on the vibratome. So yeah, there was an ongoing necrosis that was there, but I can't give you quantifiable data. Okay. All right. And just to end this first part of the talk, I want to go back to those two monkeys which didn't show any adverse outcome after PD-1 blockade. And ask what is it that might predispose an animal to exacerbation of disease upon PD-1 blockade. Now in the cancer literature, there's a lot of amazing data. It's very clear that intestinal microbiota has a major role in determining the outcome of PD-1 blockade in humans undergoing cancer immunotherapy and as well as in mice. And so we wondered if there'd be something going on with the microbiota. So we collaborated with Ranjini in Alan's lab. Now, this first graph is just to say that PD-1 blockade and TB, neither of those two things really do much to change the microbiota and in the gut. But interestingly, if we look at just the anti-PD-1 treated group before we infected them, the two macaques that didn't have an exacerbation disease we call non-responders, they actually separated. Their intestinal microbiota was similar to each other and separate from the rest of the animals. And so there seemed to be even though this is an extremely small N, there seemed to be a correlation here with the two macaques which didn't get sick after PD-1 blockade. They had the same microbiota and it was very different than the rest. And we could go in and look at the different bugs that were there. And some of them, like Ruminococcacea, have been interesting in cancer literature. So maybe the intestinal microbiota of all things has something to do with the diverse outcomes. Okay, so I think there's evidence in humans, mice, and now non-human primates that negative regulation of immune function through PD-1 is actually protective in the context of TB. And the biggest implication here is that this Nobel Prize winning immune checkpoint blockade for cancer may actually have tuberculosis as a potential adverse event. We have no idea how likely it is to be a problem. Maybe it's going to be less of a problem than TNF blockade. Maybe it's very rare. But in our macaques, it was 4 out of 6. And so we're keeping our eye on this as PD-1 blockade gets used more and more often in tuberculosis endemic regions. We're very curious as to see if there'll be a spike in checkpoint blockade associated cases of tuberculosis. We think this may have to do with the role of PD-1 in suppressing bad inflammation, but I also want to point out that we seem to have screwed up our CD4s because they didn't accumulate as fast, they upregulate CTLA4, and they didn't move as well. So it's also possible that negative regulation through PD-1 may be important for allowing the T cells to do what they're supposed to do, which is perhaps a subtlety, but to me was a really major difference in the way I was thinking about this molecule.
We have another quick question.
Yeah.
Or two, actually. So one from Kevin, is the protective intestinal microbiota associated with changes in granuloma structure, perhaps less caseation?
We haven't quantified that, but I would say it must be the case because those 2 macaques which had unique microbiota had very few bacteria in their granulomas and no necrosis. We didn't measure it, but I'm certain that must be the case.
And then one from Chetan, do you think the effect on CD4 would have been different if you blocked later, for example, effector rather than priming?
That's a wonderful question. I wish we had the money to do that experiment in late blockade. Yeah, very well could have. We wrestled hard with the decision as to when to start blockade, and we erred on the side of caution. But it's quite possible if we would have started later, we would have had a different outcome. We didn't do the experiment, so I can't say. Okay. Man, it's weird to answer a question and not tell if the person liked it or not. All right, so I want to switch to the second topic here, and so we've been trying to boost.
They liked your answer. They said it.
Thanks. All right, so we've been trying to boost conventional T cells and having no luck. So we really got interested in all of these other unconventional or donor unrestricted T cells and the potential of targeting them in the context of TB, and in particular this population of MAIT cells. And so very, very briefly, MAIT cells are a population of invariant T cells that express a T cell receptor that recognizes small molecule intermediates that are produced by microbes during riboflavin biosynthesis. And it's presented in the context of an MHC1-like molecule called MR1 that is extremely non-polymorphic, highly conserved molecule. So this is a really highly conserved pathway of recognizing non-peptidic antigens. And the idea is that the immune system is sensing bacterial or microbial metabolism. And there's a wealth of data. It's too much to sort of mention here, implicating the possibility that implicating MAITs is potentially very important in the context of TB infection. Certainly, MAITs can recognize TB infected cells. And there's a number of really important clinical correlations and polymorphisms associated with outcomes of TB. So there hadn't been a lot of data, even though there had been a lot of excitement about MAITs in the field. There really hadn't been a huge amount of data in directly addressing them in animal models. And so we were interested in following this up. And so were a lot of other people. There were actually 3 papers that were published back to back, almost simultaneously. Just a few months ago, one from Siobhan Calley's group, one from Michael Glickman's group, and one from our group, all of which tried to ask if MAIT vaccination could be important in the context of TB. And I'm going to go through our paper here. So we first looked to see if MAITs respond. Do they even make an immune response? If you infect a mouse with Francisella, the bacteria the MAIT cells go bonkers and expand 100-fold, and it's really very obvious. when we infected we looked at the MAIT cells with a low dose aerosol of Mtb infection in a mouse, they didn't budge. There was no expansion of the MAIT cells at all. But they did get activated. So if you look at the expression of different activation molecules, in particular PD-1 and CD4, both of which are induced by peptide or TCR stimulation, you can see that the MAITs are getting activated. They're just not expanding much. So the MAIT response to TB is kind of a weak one. They see the antigen, but they're not really induced to undergo expansion. And when you infect an MR1 knockout mouse, there's really no phenotype at all. So the endogenous MAIT response to TB is weak and not terribly important for the outcome of infection, but that doesn't mean that you can't make them be important. So we wanted to ask what if you had a whole crap load of MAITs at the time that you were exposed to TB? So we vaccinated mice for TB, and this has been published by others. You need to mix the adjuvant or the ligand, 5-OPRU is the TCR ligand, with an adjuvant like CpG. And I should say Sang-Mi Oh and Cliff Berry really went through a Herculean effort to make us an absolutely enormous amount of this ligand, and it was a pretty brutal chemical synthesis for them to work out. Now, if you mix adjuvant and TCR ligand, you can see the MAIT cells expand 100-fold. So they're really quite easy to make a lot of MAITs in a mouse. Now, if you do this, you expand MAITs, and 6 days later, you have this huge number of MAITs. You float 100 bacteria into the chamber and infect the mice, and then look 35 days later, there was no impact on MAIT vaccination and enhancing bacterial control. In fact, it was no different than an MR1 deficient animal. So if you have normal MAITs, no MAITs, or a ton, bacterial loads are the same. So therapeutic vaccination, prophylactic vaccination didn't seem to help outcome. And this was identical to the results seen from the Cali and Glickman labs. So we next asked if there could be a therapeutic benefit. Well, what if the mouse was chronically infected and then you started hitting the MAITs? And so we did an experiment where we infected the mice, and we waited a very long time, 140 days, and then we started giving them 5-OPRU once a week for 3 weeks. And we did as a control 6-FP, which is an MR1 binding molecule, which does not stimulate MAIT. So it does not stimulate the MAIT TCR, but binds MR1 as a control. And here we didn't even need to add adjuvant because tuberculosis is the adjuvant. Just instilling 5-OPRU into the lungs of a chronically infected mouse led to a pretty large expansion of MAIT cells, similar to vaccinating with adjuvant in a naive mouse. And surprisingly, that led to a substantial reduction in bacterial loads 3 weeks after treatment in the lungs. That it was relatively specific to lungs. We really didn't see the effect in the liver. And so this really surprised us because this is at a really late time point where the reduction in CFU is not because they continued to grow in the control animals and just didn't grow. This is a bona fide microbicidal effect, we're pretty certain. And we can't think of any other immunological intervention that would result in a log drop in bugs in 3 weeks. I mean, even injecting peptides for conventional CD4 T cells wouldn't give you anything near this. And of all things, it's MAITs, which didn't have a role in the endogenous response. So this was really quite a surprise to us. And we asked what the mechanism could be. MAITs in mice primarily make IL-17. And you can see when we blocked IL-17, we largely abrogated the reduction in bacterial loads that we got with 5-OPRU injection. And we didn't see a role for TNF or interferon gamma. And so we think this is primarily an IL-17 dependent mechanism. So that was exciting enough for us that we decided to follow up on this in non-human primates because there's really a lot of key differences between MAIT cells in mice and monkeys. And for example, in a mouse, MAIT cells are primarily CD4 CD8 double negative, and they make IL-17. In monkeys and humans, MAITs are primarily CD8 positive and make interferon gamma. So it's almost like Th17 versus Th1 kind of cells. And so we wanted to ask if this would work in a macaque. So we did a very similar experiment. We infected the monkeys, and then we waited till 6 weeks post-infection, and we started injecting them with 5-OPRU. Now, what really surprised us is 3 out of the 5 animals that received 5-OPRU developed acute symptoms, and the vets made us take the animals down early. for humane reasons. They severe cough, extremely labored breathing, head tilts as they're trying to open up their windpipe. And so we actually had way more animals in the PBS group. All of the animals in the PBS group made it to endpoint, but we lost 3 out of 5, the treated animals. So that was not looking good for 5-OPRU therapy. We did longitudinal PET-CT imaging in these macaques as well. We did many time points, but I'm just showing 3 time points here on the left. And there was really no difference. So even though we had 3 animals go down early, that wasn't necessarily because of a really huge spike in lung disease, at least as we could tell by PET-CT imaging. And that wasn't really associated with a big increase in bacterial loads either. So here I'm showing the bacterial loads again. Each dot is an individually resected granuloma, and I've separated out the animals that had to be euthanized early because it didn't seem fair to compare them. If you pull all of the animals together, there's really no difference in bacterial loads and the granulomas between the 2 groups at all. You can see in the earlier necropsies, the animals that had to go down earlier, there's a trend that there's higher bacterial loads, but we wonder if that's just a normal progression. Bacterial loads in granulomas tend to go up and then down over time. We really interpret this to say that there was no big obvious effect on bacterial loads, and that fits with the PET-CT data as well. So we had animals go down early, but we couldn't really explain it with by lung disease with PET-CT imaging or bacterial control. But upon necropsy, it was pretty clear. There were big old lymph nodes pressing against their windpipe that was impinging on the larger airways. And we could actually quantify that here. So this is just an example image of the bronchoconstriction that we saw as a result of these lymph nodes impinging on the airways. And we could even quantify that. You can see in the green lines on the right as we tried to actually quantify the percentage of percent constriction from baseline. The bacterial loads in the lymph nodes were not different, nor was the PET-CT signal. So quite frankly, we're not sure if this was bad luck or if this was an effect of the 5-OPRU, because macaques are well known to be prone to lymphadenopathy, fatal lymphadenopathy. Joanne Flynn has published a number of papers digging into the details about this. So it's possible that 5-OPRU did something to drive the lymphadenopathy, but it's also possible we just had bad luck there. But whatever the case, we certainly did not have a clinical benefit with 5-OPRU injection into the lungs of the animals. So what did we do to the mates? First we asked did the mates even get stimulated? We were very nervous about dosing and where we're going to hit the mates. So we looked at PD-1 as a marker of activation, and we very clearly saw a huge induction of PD-1 on all of the mates, not only in the bowel I'm showing here, but also in the blood. There was a systemic high-level induction of PD-1 selectively on mate cells after 5-OPRU. So we hit the mates for sure. The real surprise, in all of this is that the mates didn't expand. So we know they got hit, but they did not increase in number. So in a mouse, if you do this, you get 100-fold, 50 to 100-fold expansion in mate cells. That didn't happen in the macaques. There was really no change at all. And if anything, a drift down in the number of mates we found in the bowel. And I'll just say we looked for mates everywhere, and they didn't increase in any of the tissues. And so 5-OPRU treatment of a TB-infected macaque did not expand mates. We next looked at their function, and this is just some example fact spots for the aficionados. You can very nicely quantify mate function because you can still get on tetramer after stimulation with ligand. And so we're looking at the function of 5-OPRU specific cells after stimulation with 5-OPRU. This also reminds me to mention again that unlike mice, mates and macaques primarily make interferon gamma and TNF and a little bit of IL-17. And also we can always detect some GM-CSF. That is definitely real staining. So as we looked at the function of the mates, and so I have the kinetic going down here, you can see after 5-OPRU in vitro stimulation, in the PBS group, the function of the mates really doesn't change over time. But in the 5-OPRU in vivo treated group, you can see as soon as the week 8 time point, when we first start inoculating the animals with 5-OPRU, you can see the cytokines start to get lower in production and just be gone altogether. And so this is the summary data here. After we treated these animals with 5-OPRU, the mates very rapidly lost their ability to produce cytokines. And this is a catch-all gate for both cells that make all 4 of those cells making any of those 4 cytokines. So rather than expanding the mates after 5-OPRU injection, we screwed them up. We asked if this was true if we were to bypass the T-cell receptor with second messengers like PMA ionamycin stimulation. And we saw that the function in response to PMA ionamycin was largely preserved. So the mates weren't dead. They weren't just totally screwed up. They had a defect in the ability to respond to through their T-cell receptor and produce cytokines. So this sounded a lot like exhaustion. We're being careful about calling it exhaustion because mates have a very different history than conventional T cells that undergo exhaustion. But it, for all intents and purposes, looks a lot like functional inactivation, functional exhaustion. Now, we wanted to, I'll show you one last experiment. We wanted to ask if this was, we thought this could be due to a number of reasons. It could be due to the fact that it's the tuberculosis infection that's in a macaque for whatever reason is preventing the ability of the mates to respond. Perhaps we gave too much of the TCR ligand and this is some sort of high zone tolerance. Or maybe PD-1 is just, it's really being induced to high level. So maybe it is just shutting off mates. So we wanted to address those 3 questions in this next experiment. And so we took uninfected macaques and they were vaccinated with CpG and 5-OPRU, and we dropped the dose down 10-fold this time for 5-OPRU to try and address the concern of high zone tolerance. And then we had a second group of animals that received the anti-PD-1. So there's no TB, we dropped the dose, and some of the macaques got anti-PD-1. And just to ask again if the mates got activated, again, we dropped the dose quite a lot, so we weren't sure if this was going to still be stimulatory. But it was. You can see very clear upregulation of high levels of PD-1 after vaccination on mates in the bowel. And since we stained with the same clone of anti-PD-1 as we treated the macaques with in vivo, you can see it's blocked. So this is also a nice control to say that we've pretty effectively blocked PD-1. We've occupied the PD-1 on the surface of the mates with the in vivo antibody treatment. And so the question was, did the mates expand? They did not. So even by reducing the amount of ligand and blocking PD-1, we were not able to drive mate cell expansion in vivo in a macaque. So lastly, we looked at the function of the mates. While we still saw a drop in function after treatment, PD-1 blockade partially alleviated that drop in function. So the exhaustion that we saw wasn't because of the TB. We saw it again, even with mate vaccination with 5-OPRU and CpG. And the lack of expansion was not due to PD-1, but it was the loss of effector function was partly due to PD-1. So it was a bit complicated, but essentially what we found was that even with trying to give a bona fide mate vaccine, instead of growing mates, we kind of ruined them. Okay, so just to summarize this last part of the talk. So in mice, 5-OPRU treatment really gives spectacular mate cell expansion and drives bacterial loads down in a mechanism that we think is primarily IL-17 dependent. In macaques, 5-OPRU treatment was not protective at all, maybe even killed a few macaques through this weird lymphadenopathy we saw. But what happened to the mates is they didn't expand at all, and instead they underwent a functional inactivation that PD-1 could only explain a small part of. So in the end, the fact that we weren't able to generate large populations of mate cells means that we really haven't tested our original hypothesis, and that is that large populations of mate cells may be protective in TB. So we still don't know if we were to get mate cells to expand, if it would enhance control of TB. So we don't know that yet. But I want to put this in a little bit more context about this lack of mate cell expansion and exhaustion that we saw after 5-OPRU treatment, because I think this is probably maybe happening in humans. So I have not yet found, and I'd love it if someone in the audience could tell me an example where infection leads to a huge bloom of mates, like on the par with what you'd see in a mouse, or what you'd even see for conventional antigen-specific T cells undergoing clonal expansion. In these controlled human infections with salmonella, live vaccinated with Shigella, and all of these mate cells, if anything, go down. They don't really increase at all. Now, given that our data, you could always argue that that has something to do with the disease process, right? But in light of our data, or that the cells have left and entered tissues, and that's why they went down in the blood. But in light of the monkey data, I think we were going to speculate that, sort of typical T cell expansion that you'd normally see for other sorts of T cells through extensive proliferation is really not a normal part of the mate cell response to infection in humans and non-human primates. Mates can increase locally in precursor frequency as they migrate into a site. And so you'll find examples where mates have gone up a little bit, but it won't be hundreds of fold increases like you'd expect if this was a bona fide clonal burst, like you'd see for a normal T cell response. So I don't mean to say that mates, you'll never see more mates in your different groups of whatever clinical scenario you're looking at, but it's in larger perspective is here is those differences are going to be relatively small as compared if this was a big clonal expansion of cells. And don't really understand why that might be. It may be limited pathology. Mates are kind of a really high precursor frequency anyway, so why would you want them to expand a lot more? I don't know what the answer is as to why we saw this. And I don't understand why it's different in macaques than it is in mice, but there's some fun speculations there. Regardless, the inability to boost mate, to increase mates in vivo may represent a pretty significant barrier to therapeutically targeting mates, not just for TB, but for any kind of setting. There's a lot of interest in targeting mates in other infections and even in cancer. And so I'm not sure if it's well recognized yet that growing mates in a macaque is very different than trying to grow them in a mouse. And I think it should be a priority, at least it certainly is one of ours, to try and figure out mechanisms to overcome this poor mate cell expansion and functional inactivation. See if we can actually get them to recapitulate the mouse phenotype and ask if they enhance control of TB. So overall, in the first part of the talk, we asked if inhibitory receptors like PD-1 could be targeted in the context of TB. I think it's unlikely that will be a useful approach. And in terms of the second part, branching out into more unconventional T cell populations like mate cells, it looks extremely promising, but in order to actually test it, we need to understand how to make mate cells expand in a macaque. All right, and very quickly, all of this work was done by basically everyone in the lab. When we do macaque experiments, it's all hands on deck. The PD-1 study was led by Keith Kaufman, a biologist in the lab, and all of the mate data was led by Sake, a staff scientist in the lab, with a huge amount of help from Nikiana and Danielle and basically everybody. And all of the macaque work is a very close collaboration with the tuberculosis imaging program led by Laura Vai here in NIAID. And I guess with that, I'll stop and take any questions there might be.
Cool. So some people have multi-part questions, so they're going to unmute and ask you
Okay.
for feedback and stuff. But I have a quick question from Javed. What do you think that this teaches us about how mates work based on this data? What are they supposed to do?
Yeah. Well, I mean, in terms of what they're supposed to do, I think the best data we have is what the actual role of these cells is in modern humans is from that single report of an MR1 deficient patient. There may be more, but I know of just one patient that's been reported, and they developed a couple of different difficult to treat infections throughout life, and two of them were viral, one in the lungs and one in the skin. I think it was papillomavirus after a tattoo or something. So, in the one patient, they had an infection in the gut, in the skin, and the lung. So they seem to be clearly important in barrier sites. And not just to bacterial infections, maybe viral, their role in antiviral defense could be key as well. But I think overall, mates, they're recognizing an antigen that's produced principally by the microbiota. right? So the microbiota makes the 5ARU, which generates the ligands for mates to recognize that the mates recognize. So they're probably constitutively being stimulated by the intestinal microbiota. And I really don't understand how. I mean, there's some sort of danger signal or DAMP or inflammatory innate cytokine, which tells them the difference between a pathogen and the microbiota. But it's not clear to me why you would want a population of T cells that sees both your microbiota and pathogens. It seems like it needs to make a very important decision as to whether or not it should make a response or not. And maybe large clonal expansions is not really what they're supposed to do. You have a lot of them already. Maybe they're maybe that the number you have is elevated because they're not meant to undergo clonal expansions, and there could be a lot of important checkpoints put on their ability to increase in number. We looked at Ki67 expression, and these cells go whopping high, very bright for Ki67 expression after 5-PRU stimulation. And so they're trying to enter cell cycle. So we think that maybe there's an active, the cell cycling is being balanced with death so that the overall numbers don't change. And I don't understand what that mechanism might be, but overall, I'm thinking it's telling us that maybe you don't want lots of mates because they see your microbiota, and it might be dangerous if you had a whole bunch of pissed off mates. But that's really just speculation. I love the question you asked.
Can we ask like the direct questions now, or?
Yeah, sure.
Hey, Dan, this is Sarkar.
Hey, Sarkar. Good to hear you.
Outstanding talk, Dan.
For
the first part of the work, so there was a quick question. Maybe you showed the data, but I missed it. So was there a correlation between the extent of the adverse events upon PD-1 blockade and the level of PD-1 expression on the CD4 and CD8 T cells? I mean, also any correlation with the function of the T cells at the time of initiation of the blockade? Because, what I'm trying to get at is that if the T cells were in a somewhat functional state when you started the blockade, the adverse events were more, and if they were more exhausted or they lost more of their gamma TNF, I mean, albeit you showed that the function is predominantly IL-1 beta mediated, but they had a worse outcome if you treated them, similar to what you saw in LCMV back in your original Nature paper.
Yeah, that's an interesting point. I hadn't thought of that, quite frankly. I think trying to remember, we looked at PD-1 expression at the time we initiated treatment, and remember, we started treating at day 14. No, you know what? Yeah, okay. I'm trying to. I don't want to answer you wrong, so I'm trying to be very careful about the way I phrase it here. At day 14, there's really very few antigen-specific cells. So we've started the treatment at a time where priming is probably just starting in the draining lymph nodes. We did try to detect PD-1 expression on these cells early during blockade, but it's difficult because we tried many clones of PD-1 in all but one were blocked by the in vivo injected antibody. And so we couldn't see the PD-1 because it was masked. We found one antibody that bound a different site on PD-1 so we could still stain for it after in vivo treatment. So like at a very early time point. But the problem was it was so dim, it was almost not helpful. So I guess the answer is we tried to ask the question without even knowing it that you mentioned, but I don't know that we had a good answer. It'd be interesting if we would have waited longer, to wait for the cells to really upregulate PD-1, like the other question, and ask if that had a difference. But in the way we did it, it probably doesn't explain it because it was very early during priming, and so there's no sort of chronic stimulation. And quite frankly, Sarkar, I don't think CD4 T cells undergo exhaustion in tuberculosis like CD8s do in LCMV. CD4s are very different in the way they adapt to persistent antigenic stimulation in TB as compared to CD8s in LCMV. So it's a tough comparison to make. I'm sorry, I don't know if I answered your question. I was kind of babbling.
Thanks, you did. Absolutely,
okay.
You did. Is the background from X-Files your favorite?
Wow, good memory, man.
I know.
Well, so we have two more questions. One is from Greg. Do you know if dirty or wild mice have different MAIT cell expansion? Could the expansion be an artifact of the SPF conditions? And then Chetan's going to ask one himself after this.
I don't know if anyone's looked in dirty mice. We've wondered that ourselves often. But we've tried to do the dirty mouse experiments. Our vets will not let us do it in our facilities. And we've really bent over backwards to try and make that happen. But yes, I like this hypothesis that the microbiota is different in an SPF versus a dirty animal like a monkey or a human, in that the mates may be in a very different state in an SPF mouse as compared to compared to a macaque or human. And I think that maybe there's evidence to think that. So just looking at PD-1 levels in a mouse, the mate in a naive mouse and an SPF mouse, at least in our colony, mates are almost dead negative for PD-1. But in macaques, in an unaffected macaque in the bowel, mates are always positive for PD-1. They're not high, but they're positive. So I would suggest that maybe in a naive mouse, mates are kind of like resting naive cells. But in a macaque, they've probably been persistently stimulated for the life of the macaque by antigens derived from the microbiota. And so they've perhaps more adapted. Ron Schwartz would call this adaptive tolerance, where persistent stimulation leads to a downregulation of the threshold of activation, so that cells tend to learn to sort of live with the chronic stimulation and not undergo massive expansions when ligand availability changes dramatically. But yeah, I think it very well could do with the SPF mouse, in that all of our data, that the majority of the data in the field, and especially trying to develop mates as therapies, is done in mice. But mates may be completely different beasts for a number of different reasons in a mouse as compared to a human or macaque.
Hey, Dan.
Hey, Chetan.
Can you see me?
Yes.
I think we're at time, Robin, right? So maybe I'll just make it quick.
Yeah, we can go a little over, but you'll be the last question.
Okay. I guess the first thing I just kind of wanted to say for the graduate students is just, it's just such beautiful science. And I think, what the students watching this should take away is, like, ask big questions and go for broke. Like, you might faceplant, but you might actually make like a real discovery. And you didn't mention this, but that's kind of what you did you were a PhD student and you discovered PD-1. So congratulations. And it's just beautiful stuff. And I think you're fundamentally just right, spot on. Every time we've looked at a donor unrestricted T cell subset, whether it's MAIT cells, gamma delta T cells, CD1 restricted T cells, BCG vaccination, infection, and we're looking at sort of the traditional things we think about by virtue of clonal expansion and stimulation, we just don't see that much signal, right? It's pretty underwhelming. But it doesn't mean that there isn't something more subtle happening. And I think your point about
Sure.
Ki-67 has to be taken into consideration, because if you look back at the Salmonella papers, where they didn't see a difference in total MAIT cell numbers, but they see some activation, and then they looked at clonotypes,
Yeah.
they clearly see clonotypic expansion of subpopulations of MAIT cells. So you have to sort of ask the question, I think, at that level, not just kind of the bean counting level. Having said that, though, it really does beg the question of, like, what are they doing and do they matter? And I think here, what you've done, and Sake has done by taking us to the therapeutic time point rather than the preventive time point, really sort of just, I think, has changed my whole thinking about it, right? Because we are, again, thinking about this in a very conventional way. And I have to say, just as valuable is your positive data is your negative data, just how unimpressive it is when you try it as a preventive vaccine strategy.
Yeah.
And, one question to you is, the IL-18 signal from the first study, did you look at mates in that study?
We did. First of all, thank you. I faceplant a lot, and I totally I advocate for that all the time. But we did look at mates in the PD-1 blockade study. It had zero effect on mates. So mates don't seem to respond in macaques, and blocking PD-1 did not enhance the response in endogenous TB infection. And as you saw, what it did with vaccination. The IL-18 signal, I'm not sure if IL-18 is actually causal or not. We've done one experiment in mice, actually I guess it's 2 now, where we blocked IL-18 in a PD-1 knockout mouse infected with TB, and it didn't do anything. So we were not, Caspase-1 deficiency extends the survival of a PD-1 knockout mice, but with antibody blockade, IL-18 blockade did not. So we're going to make the knockout, and we'll block IL-1 and stuff to see and try to chase that down. But IL-18 may just be a correlate. I don't know how causal it is. It may just be reading out the inflammasome activation.
Great. And yeah, thanks. It would be so great to just have a glass of wine at a Keystone meeting and just talk in detail, right? But not today, hopefully soon.
I hope so soon too.
Awesome. Well, thank you so much for presenting, Dan. Is it okay if anyone has more questions, they can email you?
Please.
Just find if you just look up his name, it'll pop up. But thank you so much for presenting. It was really interesting, precise data and experiments. And I think that's it for the seminar.
Thank you again for the invitation. It was fun.
Yeah.
Thanks, Dan. We'll catch up sometime. Bye.
All right, guys. Bye, everyone.
Dan, bye.
Bye.